Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Harnessing Mass Photometry for AAV Sample Characterization in GMP-regulated Environments. I'm Casey Nevins, an editor at BioInsights, and joining me today is Cátia Crespo, who will explore how new compliant software solutions address all regulatory requirements, including user management, audit trails, and signing options when exporting files from Refeyn software applications. Now, I'd like to introduce our presenter. Cátia Crespo is a director of product management software at Refeyn. Prior to joining Refeyn, she worked as a product manager for leading companies. Cátia completed her PhD and postdoc at the Max Planck Institute of Molecular Cell Biology and Genetics, where she carried out multiple research projects in the fields of ophthalmology, developmental biology, and genetics. I will now hand you over to Cátia to kick off today's presentation.
Hello, I'm Cátia, and I would like to start by welcoming you to this webinar. In the coming minutes, I will be talking a little bit about the use of mass photometry for AAV analytics from R&D all the way to manufacturing, giving special importance to the new products we are launching, which will enable the use of the SamuxMP and the SamuxMP Auto in GMP-regulated environments. More specifically, today I will start by giving you a short introduction on what mass photometry is and how it can be used for AAV analytics, to show you the SamuxMP product line available at Refeyn and the performance of the instruments. And more importantly, I will be able to show you our new offering for GMP-regulated environments, which will include 21 CFR Part 11 compliant software, as well as the IQ, so the installation qualification and the operational qualification. We will also have a Q&A session at the end, and you can ask any questions you might be interested in. One of the biggest challenges in the production of AAV is that not all viruses are actually full, meaning some of them will not contain any of the genetic material which is essential for the gene therapy, but it will merely be an empty capsid. A major reason for high amount of empty capsids, even in manufacturing, is the lack of technologies to rapidly assess and determine the empty-full ratios. If we would have to say what would be an ideal technology for do an analytical measurement, we would say we want something easy, fast, accurate, and that would require low sample consumption. And these are actually some of the advantages that the SamuxMP can offer for AAV analytics. So let me show you a little bit what I mean. For those of you that are not so familiar with mass photometry is a single molecule technique that measures the molecules in the native stage in solution. During a measurement, when pipetting into the device, you'll see the molecules landing on a glass surface. So in this case, you can see here on the left, the molecules landing on the glass. And as they land, they will scatter light, which is then captured by the instrument. The scattered light is proportional to the mass of the molecules, meaning bigger molecules will scatter more light, having a bigger contrast value, and smaller molecules will scatter less light, having lower contrast value. Once the device is calibrated with a molecule of known mass, the contrast measurement is converted into mass values, and you're able to see the histogram, like is shown here on the right side of my slide, where you can identify the different populations, so the different molecules, and their masses. So in this case, you can see on the left the empty AAVs, and on the right, the full AAVs. By quantifying all the single molecules of these two populations, we are then able to establish what is the empty-full ratio. And you can see also here in between that you have some partials. This type of measurements can be done in a SamuxMP, where individual measurements take approximately 2 to 3 minutes. So 1 minute recording, and then some time for the pipetting and for finding focus on the instrument. For single measurement, you'll need to use very low amount of sample, more specifically 10 to 20 microliters of a sample with a concentration of about 10 power to the 11 particles per mil. And these measurements are quite easy and fast to do. Actually, when I first joined Refeyn, every employee that was joining the company would actually get to try to use the instrument and do some measurements independently of their background and independently of their role. So this shows you how almost anyone can use the instrument. If you're wanting to save some time, then you can use the SamuxMP Auto, which will do 24 measurements autonomously. The SamuxMP is not only easy to use, but also performs well with different AAV serotypes without any change in the sample preparation. On the left, you can see data acquired in our labs using mass photometry with 4 different empty AAV serotypes, and on the right, you can see samples measured by the Albert Heck Lab, who are experts in mass spectrometry, specifically charge detection mass spectrometry, so CDMS. They basically measured empty capsids of AAV from 2 different commercial sources and also AAV5 serotypes. And no differences in performance were observed among them. Moreover, the values of the samples on the right were compared to the values in mass obtained using CDMS, and the values obtained by both techniques were quite comparable, underlying the reliability of mass detection of the SamuxMP. A common question about the SamuxMP relates to whether the system can be used for quantification of populations of heterogeneously filled AAVs. One of the aims of Refeyn's collaboration with Pharmaron was actually to answer this question. When they analyzed a commercially available reference sample by AUC, they could identify 3 different populations of capsids containing distinct quantities of cargo. Peak A is likely empty capsid, as it corresponds to the known mass of an empty capsid. They analyzed the same sample on the SamuxMP, and as the data in the table shows, we can see that the results are in good agreement with AUC. By eye, we can also easily identify the 3 distinct capsid populations in the preparation, showing that yes, mass photometry can look into populations of heterogeneously filled AAVs. We then went and further compared the empty-full ratios obtained by mass photometry with the golden standards, basically AUC and cryo-electron microscopy. This was done in collaboration with both Pharmaron and Catapult, and you can see the results that we got on this slide. So on the top table, we have the results acquired by Catapult. On the bottom table, we have the results acquired by Pharmaron. And in both cases, the SamuxMP data is in agreement with the results obtained by the golden standards, reinforcing that the SamuxMP accurately determines the empty-full ratios across the different measurements. When thinking about where the SamuxMP is mostly used, so at the moment, it has been used for optimization of process development protocols. In this particular example, also done by Pharmaron, they quickly analyzed samples collected at different stages of the downstream purification process, from post-harvest throughout the chromatography column separation. When analyzing different elutions from the column, it is shown that the empty AAVs elute earlier, with elution 4 being the most enriched for full capsids in the particular instance. The series also illustrates that the SamuxMP provides better results after clarification. Besides protocol optimization, the SamuxMP can also be used for quality control of samples. So in this particular case, our collaborator looked at samples from the same vector produced with different manufacturing protocols. What they observed is that vector B showed lower transgene activity after transduction compared to vector A. When they put them in the mass photometer, they could actually see the reason for that. Vector B shows a large amount of partially filled capsids, while vector A was very clean. These mass photometer measurements are in line with the QC and the functional assays that they performed. But you can get to this information very quickly in just minutes. And it nicely shows that during the process development, you can quickly compare two different approaches in the mass photometer and get really meaningful information of what it is. Until now, mass photometry has only been used for AAV analytics by R&D and some process development teams. With the new launch of our 21 CFR Part 11 compliant software and also the offering of the IQs or Qs, this technology can now start being used in GMP-regulated environments. 21 CFR Part 11 compliant software package includes functionality on the user authentication and access control, also on the audit trails to enable the data traceability integrity that is required, recording who did what and when. The software package also includes electronic signatures when exporting files from the analysis application, which will contain the final AAV ratios. Besides the software, like I mentioned before, we will also offer the installation and the operation qualifications for the instrument, which will be performed by a member of our services team. Alongside this, we will provide also training and comprehensive documentation, which is essential for the use of any instrument in GMP-regulated environments. The software package mentioned will include 3 software applications. You'll have access to SamuxMP, which is installed at the server level and can be accessed through a web browser. This is for controlling user access and authentication, also for configuring some parts of the system, and to store the audit trail. I will show you a little bit how it looks in a demo in a few minutes. Then there's also Acquire MP and Evaluate MPS. Acquire MP is for the acquisition of the data and some processing of that initial data. Evaluate MPS, for those of you that already used mass photometry before, it will replace Discover MP, and it will enable a determination of the AAV ratios in a simpler and more straightforward way. You will also be able to export data from Evaluate MPS containing the electronic signature. So now I will show you how this actually looks like, and I can go through what a real user would be able to do with the system. Starting with Manage MP, let me open my web browser. The first thing that you'll be asked to do for any Refeyn application when using 21 CFR Part 11 compliant software is to log in. So I input my password, and now I'm in the system. Please keep in mind that what I'm showing you now is the view for a user that has access to absolutely everything. So not all users would see the same exact thing. Some users with the lower access permissions will be only able to see my account and help. So starting with help, you have some basic information on the software itself, and you'll have access to the user manual. You can access the main menu by going into the top right logo, or you can click on the logo of the web page and you get to the initial page. So when you go into my account, you're able to see your name, your email address, which is a unique identifier of the system, also the job title, and very importantly, the access permissions that you have. So this will tell you what you can access and what you can do with the system. Besides this, you're also able to see when you were first added to the system, and more importantly, to actually change the password. So in this case, I'm not going to change the password, but you'll be able to then change for whatever you'd be interested in. So now going back to the main page, I would like to show you how users can be managed and how you define the different permissions of those users. So when you come into the user management page, the first thing you're able to see are the users that are part of the system. So you can filter them based on active users and inactive users. So the active users are the ones using licenses and that can currently access the system. Inactive users are the ones that were part of the system once upon a time, maybe they left your organization. Because of the audit trail that requires information on users that might have left, you cannot really delete users, but you can then inactivate them so they are no longer using user licenses. If you want to add a user, you can simply click Add User, add the name, then you can add an email address, keep in mind that the email address is a unique identifier and cannot be changed. You can or not add a job title or more information, but one of the important things that you also need to keep in mind is how to give to this user the correct access permissions. And this is defined by the role. So in this case, let's say that Joanna will only be able to see the audit trail. So once you're happy and done, you can click Save. This will give you a temporary password that then needs to be copied and given to this user. When this user then logs into the system, they will be first asked to change their password before they actually go into the system to ensure that the password is going to be secret. You can see the multiple users On this page, you can also edit users. So in this case, you can change everything, being the role, the name. The only thing that cannot be changed is the email address because it's the unique identifier used by the system. If you want to define the different roles, you go into the access control page. Here you're able to see the acquire MP permissions, evaluate MPS permissions, the audit trail, and user management and settings permissions. For acquire MP, a basic user can access the system and acquire data. An advanced user can also use settings. For evaluate MPS, a basic user can do determination of AAV ratios by automated and semi-automated workflows. The advanced user can also use the manual, which is the way all the current AAV ratios are determined in Discover MP. I will explain a little bit more of what I mean by this when we go into Evaluate MPS. Besides accessing Acquire MP and Evaluate MPS, you can define if a user can or not see the audit trail, and if a user has or no access to this user management page and the settings page. Once you have already defined a role, you can simply give it to a user. However, if there's a new set of permissions that you would like to establish, you can just create a new role. In this case, I'm going to call it test, and I'm going to save it. And you can define what this person will be able to do by simply clicking on the different options. Once you're happy about it, you need to click save. If changing the page without saving, like for example here, let's add a new role. Let's call it role. Let's click save. Let's go here. But now I'm going back to user. When you go back to the access control, none of the changes that I had just made will actually be there. And that's because you need to click save to make sure that whatever you're defining is meant to and was not just an accidental click. So now if we go into settings, you'll be able to see some of the system configurations. In settings, you're able to define if you want to see an electronic signature and if you want to use the automated AAV determination fitting. You're also able to define the schema that you want to use for the semi-automated and automated determination of AAV ratios in Evaluate MPS. And I will tell you a little bit more about this when I show you Evaluate MPS. So starting with electronic signature, here you can decide if you want to use it or not. If using it, you can decide if it's going to be mandatory or if a user at export will decide if they want to use it or not. If you want to have a real electronic signature, the password required option needs to be activated because this is the one that will enforce the user to add their username and password all over again when actually exporting to add the electronic signature. If you don't care about the electronic signatures, but you would like to have the usernames on all the data exported, you can simply leave it mandatory, not have the password required, and still have electronic signature enabled. When having the automated AAV fitting enabled, this will be the first fitting by default happening in Evaluate MPS. However, you still have the options of using the semi-automated or the manual if you have permission to do so. When going to authentication, you're able to define how a user can access the system. There's two main routes that are possible. The default is Refeyn's email and password authentication process. Here, if you click, you're able to define how the passwords would have to be, how long they are, when do they expire, do they require lowercase numbers, etc., etc. If you don't want to use this method and you'd like to potentially use your Microsoft accounts, you can go into authentication method and use the OpenID Connect. So for this, you need to use certain protocols. So when talking to people at Refeyn, please do make sure to ask your IT team what are the protocols that are supported by our organization and talk to our representatives to make sure that it would actually work for you. When going into licensing, you're able to see how many licenses you still have available and until when are those licenses available. One other thing that you are able to see in the ManageMP is how many systems are connected to your ManageMP. This means that if you, for example, have 5 different instruments in your organization, you can connect all those 5 instruments to the same ManageMP, having the audit trail all in a centralized place. You can also decide to have all this divided and have one managed MP installation per instrument. That would also be perfectly fine. Just in this way, you can always use the same logins for all different instruments. If you are an AAV-optimized advanced user, or if you have access to user management and settings, you can also access the Calibrant management page. This page will be to control which Calibrants will be available in your AAV-optimized. You will understand a little bit better when I actually showed you AAV-optimized. Just keep in mind that there's already one offered by Refeyn, which is SamuxMP2, which will be used during the OQ and should be available as a Calibrant soon. And you are also able to add organization Calibrants. So in this case, if you want to add a Calibrant to use in AAV-optimized, you just click Add Calibrant, you put name, and then you put what is the mass, and you can define if it's going to be in use or not, and then you save. If in use, it will appear in AAV-optimized on the Calibrants page. If you define not in use, then you'll have it here on this page, but it will no longer appear in AAV-optimized. You are not able to delete Calibrants because, again, this information is required for the audit trail and cannot really be deleted from the system. And lastly, but for sure very important, is the audit trail itself. So here you can see the audit trail. You're able to see all the actions that were done, who did it, when they were done, and you're able to see more detailed information. For example, if you go into load and completed, you can see when it was loaded, which files were loaded, what was the mass calibration that was applied, what were the empty full ratios that were calculated, and if there are any major comments. In this case, for example, if you have one measurement, they all have a unique ID, which is going to be a unique identifier. When you want to look into the audit trail and find more information, you can do filtering to find what you need faster. You can do filtering based on the user, based on which instrument, based on when something happened, which application, meaning if it's a manage MP or evaluate MPS or acquire MP action, on the different actions, but also on the measurement ID. So this is what I just copied from the other file, and just to exemplify that, you can see every time and any time that someone looked into a certain file. Also, you're able to see when that file was actually recorded. You can decide to export your audit trail as a CSV and a PDF too. So now that I have showed you the main functionality of Evaluate MPS, I will log out. And I would like to show you a little video of how Acquire MP looks like. I don't have access to an instrument right now, so I just gonna show you a video and explain a little bit how it would happen. And then I can show you Evaluate MPS. Here you can see that the SamuxMP is a benchtop device. You have it connected to a computer, and to do a measurement, you just open the lid, and then you can see this little silicone gasket, and you can pipette your sample into one of these little wells. Once you have pipetted, you can close the lid, and then after finding focus in the system, you can just click record. Once recording, you start seeing the label of the molecules, and on the right, you're able to see the molecules that are being detected. So you already here see on the right side your empty and your full peak being detected. Keep in mind that I speed up a little bit this video so that you don't need to watch the acquisition for an entire minute, but this would usually take approximately a minute. Once the recording has been finished, you'll see the pop-up of the saving dialog where you can put information as the name of the sample, but very importantly, you need to put the size of your empty full capsid in order to use the automated determination of the AAV ratios. Once you have saved your files, you can then determine the AAV ratios using Evaluate MPS. So let me show you what I mean. I'm now going to launch Evaluate MPS. Please keep in mind that this is the development version, so it has this terminal window, but you would not get a terminal window like this if you're using the final installation of Evaluate MPS. I will just minimize it so that it's not disturbing us. And now the first thing that is going to happen, just like for all the other applications, is that we're going to be asked to log in into the system. So here, let me add my username and password. Here you saw that I wrote pharma wrong, and when trying to log in, the system will not allow me, saying that there's an error in the domain. So here, let me write pharma correctly, and now trying to actually log in. Once logging in, there's very little you can do in the application besides opening the data. You can go into the About option, where it shows you a little bit what this application is and what is the version that you're currently using. But the most important thing is actually to go and load the data you're interested in. So here I'm already in the folder that I want to. And the first thing I will see when loading data is the calibration page. In this page, you can see the list of all the measurements that you just opened, and you can see the list of the calibrants that you have available. So in this case, because I have written down that this measurement was mass Franz P2, the system automatically detects that it's mass Franz P2, and it shows it has a calibrant. If you have more than one calibrant, you could also select multiple files, and you're able to choose which calibrant does this contrast is associated to. So in this case, you could put something else, but because I actually don't want to use this, you could leave it unassigned. If, for example, you would have detected the peak in your calibrant measurement, but you don't want to use it, you can just leave it assigned. Or in this case, because this is not actually a calibrant measurement, I'm going to unassign it. You can also add comments if you're interested in. And once you're happy about the calibration, you can simply click Accept and Continue. And this will by default show the automated version. So you can also add comments, but in this case, I'm just going to accept and continue. If not wanting to do the automated, you can go and use the semi-automated. In this case, you're asked to choose your empty peak, you're asked to choose your full peak, and then by default, you will have your partials be in between your empty and your full. You then ask also if you want the overfull region. If you click yes, you'll have every count above the full region being defined as an overfull. Keep in mind that Evaluate MPS basic users will have access to the automated and the semi-automated workflows that I just showed you. In the semi-automated workflow, what's happening is that when you click on the peak, there will be a Gaussian fitted. And then from the center of the Gaussian, you will have, in this case, a 3 sigma from the center and a 3 sigma to the other side, because 3 sigma is what is defined in Manage GMP configurations page. We advise you to define what is a number that best works for your organization and your samples, and to stick to it, and to not change it frequently. If you are an advanced user, you can also use manual mode. So here, let's just accept and continue. And this last sample, I'm going to use manual mode. In the manual mode, you can also activate overfull, but something that you can do is also to simply drag the limits, or you can use the arrows to change it. Once you're happy with it, you can then accept and continue. Once you have accepted all the different samples that you have measured, you're brought to the final results page. So here you can see in the AAV ratios, figure type, the table showing all the different measurements. What is the empty, the partial, the full, and the overfull if you'd have calculated it. You can also see the counts. For the counts, you can see not only your empty, full, partial, overfull counts, but you can see everything that was below the empty and everything that was above full. So if you have selected overfull in our semi-automated, you will always have 0 above region because there's nothing else there. We have already accounted for everything in the overfull. You might also have situations like in the first measurement that I did, where you have no overfull because it has not been activated. And for the current automation that we have available in the system, it only enables empty, partial, and full calculation. Besides the AAV ratios, the counts, you can also see the histograms. Here we depict the different intervals and all the different empty full peaks. You can also see all the comments that you might have added during the acquisition and the analysis being on the calibration page for the master P2 or during the analysis for the different samples. You're then able to see some quality metrics, so total binding and unbinding events, but also the mean of sharpness and brightness. And it's important to notice that here, for example, in the sharpness, you always have 2 point something. And the fact that all these values are along the same numbers shows you that these measurements are good. If you all of a sudden would have, for example, a 5 or a number that is completely different from everything else, then this would be a sign that something might be wrong with that measurement. You can also see the mass calibration that you apply to all the different measurements and the metadata, which includes the measurement ID if you ever want to check for it on the audit trail, but also you can see how were the AAV ratios determined, automated, semi-automated, or manual mode. You can also see the empty capsids and the full capsids mass and sigma. Once you have looked at your data and you simply want to export a report, then you can click export. Here you can choose what do you want to export, and you can choose to export individual files all within a zip, or if you want to export a final PDF report that includes everything. So in this case, I'm going to go for the PDF report. I'm going to put it on my desktop. And because I have the electronic signature active, here I'm asked if I have any other comments. Just for the sake of showing how it looks like, I will write no comments and I will continue. Because I have enabled electronic signature with the requirement of the password, this will ask me again to log in. So here, let me type it down again. You need to log in with the same user that has been already previously logged in. And once doing it, you will then export your file. So now let's look at how the report looks like. So here you get a first page that you just have, analysis results written. And then you have all the individual figures and all the individual tables that you already had before. So you can see the AAV ratios that were calculated, also the counts. You can see the different histograms. This image has incredibly high quality. So if you ever want to truly zoom in, you'd always be able to see how these histograms look like. You're also able to see all the different comments, the quality metrics, the mass calibration, and of course the metadata. But very importantly, if you focus here at the end of every single page, you have the signature. You have the meaning of the signature. If you want something a bit different has a meaning, you can also add it as a comment. You have the name of the person. You have their unique identifier, which will make the whole signature always unique, which is the email address. You have the date and you have the time. So now that I have shown you how the software looks like, I would just like to summarize very briefly what we talked about today. Mass photometry is an analytical technique that allows for easy, fast, and accurate measurements of AAV samples using very low sample amount. This is now not only possible to do for R&D and process development teams, but also for manufacturing teams that require 21 CFR Part 11 compliant software. We are also providing installation and operational qualifications to validate the system and any training and documentation that would be required. And now I'll be happy to take any questions you might have.
Thank you.